Good morning, everybody. Welcome to our exercise physiology interest group discussion. A couple of housekeeping things to take care of. This is our leadership team. We have Michael C., Stephen Malin, one of our talkers here, Robert, Sam Scott, me, I'm one of the advisors, and early career representative is Maria, and Lisa Chow has been with us forever, and she's very helpful. Congratulations to the Exercise Physiology Early Career Abstract Award winner, Eleonora Maria Aiello. Come up and get your certificate. For today's talk, the Professional Interest Group Discussion on Exercise Physiology, Lifestyle Interventions, and Cognitive Function. Our two speakers today are Mark Espeland and Steve Malin. So Mark's going to talk about the impact of exercise on cognitive function, and Steve will talk about the implications of brain insulin sensitivity and exercise for cognition on type 2 diabetes. This is what I would consider to be a very hot topic these days. Some people would say, why is diabetes related to cognition? A lot of people would call it Alzheimer's disease type 3 diabetes because it's strongly related. And as you can see, this is taken from a recent review article. As people get older, their physical fitness declines, and with that, there's also a decline in cognitive function and increase in dementia. And there's a lot of factors that are related to this. I call it acquired risk factors, such as diabetes, hypertension, insulin resistance. There's also risk factors that aren't acquired, like genetics. So as you get older, people tend to train less, become less fit. You have a lower VO2, you have impaired amyloid beta function, the neurons are not firing quite as quickly as they used to, and there's a strong connection between insulin resistance, particularly vascular insulin resistance in the peripheral and also in the brain. So as you can see, as you get older, aortic stiffness increases, vessels are less pliable, the white matter in the brain is less functional, you have a decrease in cerebral blood flow, and your fitness also decreases. So it kind of goes hand in hand, but not all is lost because exercise training can help preserve cardiovascular fitness and also help preserve cognition to a certain degree. So a little, very brief background. You have insulin that binds to the receptor, and what should happen is a cascade of events that lead to vascular relaxation and GLUT4 translocation to bring glucose into different cells. When you have a system that is like an activated endothelium where there's an unhealthy environment, something goes wrong and the cascade goes a different direction. So you have an increase in endothelin 1, which causes vasoconstriction, cell proliferation, and hypertension. So all together in the same molecular pathway. Now, how exercise can actually help, this is a study that was done a few years ago, is exercise training tends to increase insulin sensitivity. You can see that glucose at every time point after training is lower, but insulin's the same. So insulin sensitivity is improved, glycemic regulation is improved, and even advanced glycation end products are improved with exercise. All these factors can contribute to the increased risk for dementia. This is a little bit busy, and it's the last one I have, I promise. This one talks about vascular systems. So these are people with diabetes, which is a huge risk factor for Alzheimer's disease. Having type 2 diabetes actually increases the chances of getting Alzheimer's by threefold. And about 80% of all people with Alzheimer's have insulin resistance. So if you look at before the resistance training study, there is, let's see if I can, no. Okay. Before resistance training, you can see that microvascular blood flow does not increase postprandial, where afterwards it does increase postprandially, which is important because the increase in blood flow to the muscle can help alleviate the high blood glucose. There's improved glycemic control, improved strength. So exercise does help a lot of things beyond just what you would think of as hypertension. Okay, so with that in mind, I'm going to keep my talk brief and then say, let the experts come and take over. Okay, let's welcome Dr. Mark Espeland.
Well, thank you. I'm really honored to be here, and something near and dear to my heart. I appreciate the opening introduction that really got us grounded into the field. Happy to take photographs if you'd like. I do some committee work with industry. I'm involved with quite a few grants right now and have research funding from the Alzheimer's Association. So what I like to talk about in general, exercise is good for brain health, and cover that a bit. And I was asked to focus some on executive function, so I'll do that as well too. I'll do this generally, and then I'll do it for individuals that have diabetes and some aspects of prediabetes. And then I want to look at exercise combined with other interventions, exercise combined with weight loss, exercise combined in the context of a multi-domain lifestyle intervention, and a brief thing about exercise combined with incretin-based therapy. So I think generally we all know, I think, that exercise can benefit the brain. This is a recent article from the Wall Street Journal that just sort of trumpets that as well, too. Among the different targets for interventions to improve brain health, exercise is one of the leading ones. And it's also incorporated into recommendations from research groups and clinical care groups. First one here is from the American College of Sports Medicine. It says that there's a moderate to strong support that physical activity benefits cognitive functioning during early and late periods of the life and among individuals that have some cognitive deficits. And also you see a second recommendation here that both acute and long-term moderate and vigorous intensity interventions improve brain structure and function, as well as cognition. And this was older, about 10 years ago. The National Academies put together a group to look at what recommendations could be done to the public about preventing dementia. And this panel, I was honored to be part of it, picked out 3: cognitive training, blood pressure management, and physical activity as being the 3 that had the most scientific evidence base for benefit and could be recommended generally. And we also noted that a multi-domain intervention, including physical activity, may accelerate benefits. And I think many people have seen these diagrams. They come out regularly in the Lancet by a commission that's focused on looking at modifiable risk factors for dementia and Alzheimer's disease. And this is the latest one that came out a few months ago, that these experts project that about 40% of global dementia cases could be prevented by targeting modifiable risk factors. And they break this up into those that might be important earlier in the age, those important later in the age, and among within the list, they claim that physical activity, if modified, might benefit or provide or reduce 2% of the cases of dementia, and that is. Overall, they also looked at cases at different places in the country, and they find different cases in the world. They find that physical activity is an important modifiable risk factor wherever you look. It may vary depending on culture and other aspects of the population. So. How does physical activity impact the brain? Well, some of this was covered by Ryan. There's evidence that aerobic exercise can increase brain volumes, particularly hippocampal volume. There's evidence that it can slow cognitive decline, increase cognitive resilience, and lower dementia risk and all-cause mortality. And these are fairly well established. And there's a very robust pathway whereby exercise may improve brain health. I'm not going to cover this, but just to demonstrate that in some sense you can think about physical activity as being itself a multi-domain intervention in that it influences many underlying mechanisms for cognitive impairment and dementia. And there are a remarkable number of meta-analyses that have been published looking at that. It's probably a situation where one could do a meta-analysis of meta-analyses. This one found 80 different randomized controlled clinical trials. You can't read them, and I can't read them either, but did a very systematic assessment of what could be learned from this. And in general, their conclusions are that overall there's a modest effect on cognitive function. This is clearly be a clinically significant effect of 0.2 standard deviations or so. Based on their analysis, they felt like the more intense and long the exercise intervention is, the more likely a benefit would occur. And from their review of the literature, I don't know if I agree with this, they feel that exercise was a little bit less effective among women compared with men. Now, that was healthy individuals. And there's also similar in a number of meta-analyses looking at the effect of exercise intervention among individuals that have cognitive deficits, cognitive impairment, or cognitive diseases. And in general, they find benefit as well, too. This meta-analysis looked at 19 different studies and came up with 1,100 different individuals and found a benefit of 0.47 standard deviation, so even larger from their analysis than seen for healthy individuals. So I want to focus down specifically on executive function as well, too, and it's a loosely defined cognitive domain. You see some of the contributors to it, working memory, flexible thinking, and so forth. It's very much involved with planning and organizing one's thoughts. And as you can see, with it covering a number of different abilities, no one single cognitive test really can reflect fully what executive functioning is. And so oftentimes there's a combination of different tests that focus on different underlying constructs of executive function that's used and loosely labeled as an executive function. And there's a fair amount of evidence that physical activity interventions not only benefit executive function, but perhaps benefit a little bit more than other cognitive domains. Here's a large meta-analysis that focused on executive function. I'm just showing the part of the graph that looked at older individuals. But what the authors see here is a fairly marked benefit in executive function among older adults, and perhaps more larger than compared with younger adults. And this is an old paper by Kramer and Colcom. And what they looked at in this paper is comparing the impact of physical activity on different domains and cognitive domains. And I think what's striking in this paper is that the benefits seem to be much larger for executive function than for some of the other domains. And this might be in the sense that executive function has more targets involved with it. It's got quite a few different aspects and abilities, and perhaps somehow additive function of exercise might improve it more. I want to switch now to focus on specifically on cohorts with diabetes and some extent with prediabetes. And there's a number of meta-analyses also in these cohorts, not as many, but what you see here is 4 different clinical trials that were included in this meta-analysis, and they looked at the effects on cognitive function both during the trial or at the conclusion of the trial or at the subsequent follow-up. And I guess one lesson from this is you probably don't want to work with me on these things. You can see that the trials I was involved with didn't have much benefit to say, but when combined with other well-run trials, these authors found a fairly marked benefit both during the trial and after the trial. And other meta-analyses similarly find benefits in cohorts with diabetes. In this graph, the left side are benefits from the intervention, and you can see that there's a fairly robust improvement associated with exercise interventions on these clinical trials focused on a measure of executive function. And this is a recent paper in diabetes care that looked at people that had and didn't have diabetes and whether or not they were active or sedentary. The top curves are people without diabetes, and you see that overall they tend to have better cognitive functioning compared with people that do have diabetes. But the difference between those curves are those that are inactive versus active. And of course, folks that were more active had better profiles of cognitive function. But you see something very similar within a cohort of individuals with diabetes as well, too. Of course, they do a little bit worse overall compared to people without diabetes, but even within diabetes, it seems like there's some benefit of just not being sedentary. And there might be some evidence that physical activity interventions might actually benefit more people that have diabetes than don't. This is a slide from the LIFE study. This was a cohort of older individuals that had mobility impairments, and the physical activity intervention that involved aerobic training, strength training, and balance and stretching was designed to increase their mobility and prevent mobility disability. In a secondary analysis, we looked at folks in the cohort that had diabetes. That was about 20% of the cohort, and those that didn't. And what we found on the far right there is executive function, but found some benefit on folks with diabetes, but no benefit at all for folks that didn't have diabetes in this trial. So that gives you some hope that within a cohort of people with diabetes, that the physical activity interventions might be particularly beneficial. So I'm going to switch now and talk a little bit about what one might see combining exercise with other targets of lifestyle. And I'll start with looking at exercise combined with weight loss. And so this will be applicable to folks that have overweight or have obesity, which of course is most people with type 2 diabetes. And here it's a little disappointing. There's been 3 large, well-powered clinical trials, 2 conducted in pre-diabetes populations, and the third in a cohort with established type 2 diabetes. And these were all effective lifestyle interventions that improved physical activity and resulted in sustained and clinically significant weight losses. But none of them found any overall benefit in cognitive function. And so that's discouraging because you'd expect that if you want to do a lifestyle intervention for our cohort, you would combine it with weight loss. But it's unproven, I think, that a combination of weight loss and physical activity within the context of diabetes actually benefits cognitive function. I'm going to show a little bit more data from the third study there, the look-ahead trial. These were folks aged 45 to 76, and they all had overweight or obesity and all established type 2 diabetes. Otherwise, they were generally healthy. And what we found across all the individual domains we looked at the conclusion of the 10-year lifestyle intervention, there was no evidence of benefit on any of these domains. And you can see executive function is in the mix there. So that was very, very disappointing. It's not to say that there isn't evidence in that cohort that exercise is beneficial for the brain. In the look-ahead trial, we did graded exercise tests at baseline and several times through follow-up, and got a measure of cardiorespiratory fitness from that. And both the level of cardiorespiratory fitness and the change in fitness over time were strongly related with cognitive function. And so there's a bit of a mixed message there, but it appears that if one is able to maintain fitness or increase fitness in this cohort, there might be some beneficial for cognitive function, that the lifestyle intervention that was administered in the trial just wasn't perhaps of a dose sufficient to make these changes. And there are also, within the look-ahead study, there tend to be some differences in whether or not the intervention might benefit people depending on their obesity status. In this slide here, the dark bars are individuals who came into the trial with BMIs less than 30, so they had overweight but not obesity. And there's some evidence in this post-hoc analysis that there was benefits for this group, but the shaded gray bars are people with obesity, which was the bulk of the cohort. There didn't seem to be any additional benefit. So I want to switch now and talk about physical activity in the context of a robust multi-domain lifestyle intervention. And I'll look at trials that have been completed, and ongoing trials that look at exercise combined with nutrition, cognitive stimulation, and risk factor monitoring. And these are all domains that were suggested as possibly beneficial at different academies, meetings, and so forth. The Worldwide Fingers program is a network of in 60 different countries now of these clinical trials looking at multi-domain lifestyle interventions and whether or not they might benefit cognitive function. And the trials are sort of loosely harmonized and are all committed to sharing results and networking. In the United States, the trial is called the U.S. Pointer Study, and we'll be presenting our data, our primary results at the Alzheimer's meetings next month. But some of the trials that have been completed, this is the finger study that really sort of started out the Worldwide Fingers Network. It was done in Finland. And as you can see, it's a multi-domain lifestyle intervention with nutrition and exercise and cognitive training and vascular monitoring. On the right panels are the cognitive testing scores that emerged from the trial over time. You can see that they increase. Some of this is probably attributable to a practice effect. In healthy cohorts, it's typical that cognitive tests improve over time, at least initially, once people get used to doing them and are more comfortable and perhaps less stressed. If this cohort was followed longer, these curves would bend and the cognitive aging would overcome the learning effects. But what you see here is that the control group was basically just an education type of intervention. Scores improved there, but they improved more among individuals that underwent this lifestyle intervention over a couple of years. And on the right upper panel is executive functioning, and you can see that it appeared to be fairly sensitive to the trial as well too. There's been some efforts to try and parse out, is it the nutrition, is it the risk factor monitoring, or is it the exercise and so forth that's most beneficial? But they're all entangled in the intervention, so it's hard to dissociate what is most effective. This is another trial from the Worldwide Fingers that was just completed recently in Japan, and it targeted the enrollment of individuals with diabetes or with hypertension. 23% had diabetes at enrollment. And it's a very similar type of intervention to the fingers. It focused on 4 different domains, and these are the cognitive function test scores. And the trajectories look very similar to finger. And so this suggests that these multi-domain lifestyle interventions might especially be effective in individuals with diabetes, or at least the results, the benefits from them extend to people that have diabetes. I probably would be amiss if I didn't say something about incretin-based therapies, but there's really not too much to say in that exercise is recommended as a course to go along with these to prevent sarcopenia and bone loss and such. So there's really not a case to be made that, at least in humans, that physical activity is additionally beneficial, but there is some mouse work that suggests that the effects are additive anyway on cognition. So what I'd like to leave you with are a few points that, in general, populations, higher levels of physical activity are associated with better cognitive function and executive function, and certainly it would be expected to benefit from them, and perhaps more so. And interventions that increase physical activity are likely to lead to cognitive benefits. Among individuals with type 2, it appears that the benefits of physical activity may be at least as in general populations. Combining physical activity with weight loss, however, doesn't appear, at least based on some large trials, to benefit cognitive function. However, there might be some subgroup differences as well, too. And combining physical activity with other lifestyle targets appears to benefit cognitive function, including executive function. So thank you very much. I'm happy to take questions.
The floor is open for questions. I'd like to start with a question, if you don't mind. I know that there's been a lot of exercise studies on diabetes, and there seems to be a dose response with exercise intensity and even multiple modes, and the impact that has on HbA1c. Do you notice the same effect on cognitive function or executive function with a dose response for exercise?
In the context of diabetes, yes. The recommendations from the meta-analyses are that the more intense exercise programs, the larger the potential benefits might be. So I think that would be expected. And we all know that more intense interventions are beneficial. Many risk factors are related to cognitive function, such as you mentioned, hemoglobin A1c, but they also lower depressive symptoms, improve blood pressure control, and have a lot of other benefits. Thank you.
Hi, thank you so much for your presentation. I had a question kind of related. So I know that multiple forms of exercise can be associated with better cognitive function as we age, but I'm curious, especially seeing that there's a difference between there's like a increase in that benefit in diabetic patients, if that is due to just general physical exercise, or if there's also a relation to the amount of muscle mass and different things like that as well.
Yeah. I don't know of a study looking at changes in muscle mass versus cognitive function in the context of an intervention. That's not to say there may not be something, but I think in general, the sense is that aerobic exercises may be more beneficial than strength training. There seems to be a variation in terms of the effect of the exercise depending on adiposity as well too. It may be more beneficial for people that are less heavy than those that are very heavy.
Thank you.
Hi, thank you so much for presenting your work. It was really interesting. As a neuropsychology student, I have a few questions here, I guess, but I know there wasn't a huge change with memory specifically, and there is some evidence that insulin can be protective for memory, if I'm remembering that correctly. But were all of the type 2 diabetic patients, were they on insulin, and were some not on insulin? Did you see a difference perhaps in the memory domain between those?
Yeah, in the studies I've been involved with, insulin as a marker of a more poorly controlled diabetes tends to be associated with poor cognitive function. That's not to say that there are insulin receptors throughout the brain, and insulin is important for cognitive function and so forth. And there are some clinical trials looking at intranasal injection of insulin, where it goes directly to the brain. There's some evidence for benefit there, but in general, folks that are need and require insulin are probably on a more steep pathway for cognitive decline than those that don't.
Makes sense. Thank you.
Question in the back.
Hi. I just have a question about, like, what, if the patient is asking what's the least amount of exercise they can get away with or do for benefits, do you have an opinion on that based on the studies? Twice a week, 3 times a week, 50 minutes?
Yeah, well, I think the general sense is more is better, and more intense is better as well, too. In the studies that involve with those people that are most adherent, and meaning that they meet or come close to the goals for the exercise intervention, those people tend to improve and get more benefits. There might be some circularity in that people that are able to adhere to the intervention may have better cognitive functioning to begin with, but that's what we see. So most of the interventions that I've been involved with target around 150 minutes per week of moderate to vigorous exercise.
Okay, thank you.
Okay, if there's no more questions.
Yeah, well, thank you very much. Thank you, Rand.
Okay, Steve Malin.
Hi, I'm not Gladys, as you can tell. Unfortunately, she wasn't able to make it last minute, so I'm stepping in for her. And we appreciate her time and consideration of this, and we hope to perhaps see if she'd be willing to participate in a webinar coming up this year. But with that said, we are really excited to share some information that we've been working on, and I think Mark did an amazing job highlighting a lot of topics in this field related to exercise and the potential benefits. We're going to take a little bit of a narrowed view and focus a little bit on insulin and how insulin might be partly regulating the brain, as also Mark was highlighting at the end with some recent trials. I don't have any disclosures except to say I asked for some slides not to take pictures. These are actually part of a dissertation study that are currently under analysis right now in preparation for this talk. So I thank you for that. As Mark highlighted, really, Alzheimer's disease and related dementias are really reaching epidemic proportions. Close to nearly 7 million people in the country above the ages of 65 are considered to be in this category. And, partly this is concerning because Alzheimer's disease itself has been linked strongly to the vascular system, as Ryan and Mark were alluding to a bit, and really helps implicate then the brain and vasculature in unison, almost this axis, if you will. And thinking about this, then, we have major public health ramifications for people not only with diabetes, prediabetes, but in general, as people age, related to some of the topics, including processing speed, to add to Mark's. And I think one of the interesting observations in recent years has been the identification that many pharmaceutical trials that have been conducted have been successful in implementing them. And there have been some benefits towards cognition when targeting amyloid beta, which tends to be the primary mechanism proposed for this cognitive decline. But these trials often aren't as effective as hoped. And the cost of these pharmaceutical agents may not necessarily justify implementation in the clinical realm, leading one maybe to hypothesize something else may be actually the underlying mechanism and maybe not necessarily amyloid beta as primary. So hinting at this a little bit earlier was this idea that vascular dysfunction seems to coincide pretty strongly with declines in brain health. And when I say vascular function, we tend to think about the periphery a lot and skeletal muscle as a primary depot for glucose in the regulation of diabetes control. But notice here that this aortic stiffness, so if we think about this within the body, this tends to track pretty well early on, maybe even arguably a bit before some of the declines in cerebral blood flow or structural damage in white matter hyperintensities. And interestingly enough, in kind of the topic of our session of exercise, cardiorespiratory fitness is declining over one's lifespan in conjunction with these other parameters. really raising an idea again that maybe the vascular system is a connecting highway from brain to body. And for us, starting to think a little bit about this idea from skeletal muscle insulin resistance, how does insulin fit into the brain in this capacity? Does it travel to the brain just like we would anticipate it traveling from the pancreas to the skeletal muscle? What does that all look like? And, we think there's some justification for this is, in many ways when thinking about treating diabetes, we may be really just hitting the tip of the iceberg, given the underlying pathophysiology is much more complicated with, perhaps abdominal obesity in particular being a primary culprit. But, in some ways, we tend to think about insulin resistance at the center of this, given that insulin resistance can develop with really just lack of physical activity, sitting long periods of time. So if you want to get up and stand, feel free. Seriously? No. But just saying, that may actually be a precursor, right? Initially happening where there can be a host of issues, as one might call a metabolic syndrome effect, including hypertension in the vascular system. So with that in mind, many people with type 2 diabetes are afflicted by Alzheimer's and Alzheimer's-related dementia. And it's thought on some levels then that the peripheral effects, ranging from the vascular system and the three musketeer of liver, muscle, and adipose to the pancreas, dysfunction within these may, in some way, maybe through insulin, act to actually impair the brain. And this shouldn't necessarily be surprising. We know that insulin has major ramifications for appetite regulation, being an anorexic type, appetite suppressing hormone. Insulin also is quite potent at activating the sympathetic nervous system to stimulate postprandial thermogenesis. So we often think about this thermic effect of food. But in that way too, you might be a little skeptical still, thinking, well, I was taught the brain is primarily GLUT1 dependent. Doesn't really need insulin for glucose uptake. But as Mark was pointing out a bit, that's not necessarily true. There are multiple regions throughout the brain that have GLUT4 transporters, just like skeletal muscle. And insulin plays important roles then in neuronal metabolism of glucose. And you can see here is, perhaps just for timing, a part of our talk today, the hippocampus is rich in insulin receptor density, suggesting on levels insulin might have some really important roles. Now, in order for insulin to get there, we have to identify something that's important, which is the blood-brain barrier. And this is something we don't necessarily maybe think about as in skeletal muscle. But here the blood-brain barrier actually plays a critical role, allowing endogenous insulin secretion to cross into the brain from the body and act on neurons to have a host of effects, one of which is actually helping degrade amyloid beta. But something worth noting and just hypothesizing a little bit from Mark's fascinating talk was this notion that obesity itself may have some responsiveness to interventions or blunting effects, if you will. And what's been noted in the literature is as one ages, hyperinsulinemia often develops, and in turn, that actually impairs the blood-brain barrier, meaning as one ages, less insulin is actually entering into the brain, creating a hypoinsulinic effect. Now, this in of itself could cause dysregulation or create what one might say is brain insulin resistance or neuronal dysfunction. And this is problematic because insulin, as it does enter into the brain, has a host of effects, one of which is regulating peripheral vascular tone and helping regulate cardiac activity, as well as the delivery of blood flow to tissues like skeletal muscle for uptake and utilization. So in this way, the effect of endogenous insulin getting into the brain is quite clinically and physiologically relevant. So to narrow in on exercise a bit here, we could ask a basic question, just what does exercise do to the brain? Do we know how exercise actually, in humans, impacts brain insulin signaling? And to be honest with you, there are not many studies at all on this topic. This is one that was published a few years ago by Stephanie Coleman Martin Henney's group, which was really a nice study. It was 8 weeks of aerobic exercise, about moderate to vigorous intensity. And they had found that when using intranasal insulin as a model to directly provide insulin to the brain, that cerebral blood flow was improved in response to that intranasal insulin following 8 weeks of training, suggesting on a level, at least within the putamen, that exercise is able to improve brain insulin sensitivity. That's really it. Using an intranasal insulin approach. Almost had you. There's one other study I'm familiar with, and I find this quite interesting. They used the clamp approach, which many of you are familiar with and conduct yourselves, is where we infuse peripheral insulin into the system. And in this case, the assumption is that insulin will get to the brain. And they used PET tracers, or PET imaging and tracers, to look at brain glucose uptake. And they did this then during the clamp procedure to understand, well, what effect does insulin have on brain glucose uptake and how does exercise modify that? There's some data suggesting that actually people with obesity in response to insulin infusions actually take up more glucose than a lean healthy control. And that's largely thought to be a compensatory mechanism, given that in the lean individual, there's declines in brain glucose uptake during insulin. Saying that, the responses here are put into that context then, right? When we look at this is sprint interval training, not sitting, but sprint interval training. So a high intensity exercise program. Again, in line with what Mark was saying, that intensity here seemed to be the exercise model that actually reduced brain insulin-stimulated glucose uptake. Moderate intensity exercise or moderate continuous exercise did not. What's fascinating is both groups from peripheral glucose infusion rate calculations improved insulin sensitivity, suggesting almost a disconnect between the body and the brain in this way. I bring this up because, again, there really are not many studies on this topic. And in some ways, you can even say, well, they're not even really addressing insulin signaling in the brain. They're more or less functional outcomes, which assume the signaling is intact or processing. Recently, we had the pleasure of meeting and collaborating now with Dimitrios Kapagiannis at the National Institutes of Aging. And we've been fascinating over the last 10 years or so now with extracellular vesicles and the roles that they may have in health. In this case, you can actually label proteins to identify neuronal extracellular vesicles. And these are cells that essentially are released by various organs, adipose, muscle, endothelium, brain. And by identifying them, you could actually then lyse the cells to understand the cargo or the contents within. So in some cases, people have suggested that extracellular vesicles are essentially liquid biopsies. And there have been some data showing that altered neuronal extracellular vesicle insulin signaling is associated with Alzheimer's. Given that knowledge, we were interested in thinking, can training modify these extracellular vesicle insulin signaling? We conducted a 2-week trial, so we had interval exercise versus continuous, and we had measures done both before and after. People in this study all had pre-diabetes defined by ADA criteria using an oral glucose tolerance test. And based on results of this study, we were able to look at blood samples during the oral glucose tolerance test, both at 0 minutes and 60 minutes, to identify if exercise impacts these cells in the fasting state, but also upon glucose ingestion. Overall, just to show you that the training program really had negligible effects on their body weight, as we would anticipate with being a 2-week program, but it did have somewhat relevant effects on their fitness level, suggesting here the exercise was working. When looking at glucose as well, it's important to recognize that we saw glucose tolerance benefits in patients, both in the fasting state as well as postprandial, again, highlighting over here that insulin sensitivity was improved overall. For timing purposes, I'm not going into all the specific outcomes, but knowing that, it seems that this exercise works such that if we look at the brain, perhaps there should be an effect. And I apologize for the small details here, but to point out with you, the one protein that really stood out was AKT. We didn't see any effect of exercise on the fasting state AKT levels from these cells, but we did upon glucose ingestion. And that was really interesting to us, that this glucose-stimulated effect had some relation. And when we looked at relationships between insulin resistance calculated by HOME-IR, as well as the simple index of insulin sensitivity, we actually saw the changes in insulin resistance or insulin sensitivity had correlated with AKT, suggesting here a relationship between the body and the brain. And this matches literature. So collectively here, we might say that, okay, exercise seems to be a potent way, or at least a way, to improve brain insulin sensitivity. But again, for us, at least in our study, the changes at the AKT level doesn't necessarily mean they're functional changes. So we really need to be careful on how we interpret this, but also really begs the question of, can exercise improve the way in which the brain responds to insulin? And also, does that improvement in brain insulin action, if it does happen, have differential effects in the periphery? So when thinking about how to do this, we appreciate the literature could suggest a multitude of ways, which overall here could be through neuroimages. You could then either give infusions, you could give intranasal insulin, or you could feed people, all to try and stimulate this approach. And, in part, led by groups at Wake Forest like Laura Baker, Suzanne Kraft, really highlighting there aren't many standardization protocols on this. So tackling this is a bit complicated. But the intranasal insulin approach is really intriguing to us. This was a study done by Novak's group, highlighting that in particular here, by giving intranasal insulin as a therapy, so not insulin injection as we might classically think about, but actually intranasal insulin spray. Overall here, a summary of their 24-week trial suggested in people with type 2 diabetes in particular, not only were they walking faster, but they had improvements in brain blood flow in the fasting state, and they had decreases in insulin resistance estimated by HOME-IR. And the controls did not have as many of those effects, but again, they were relatively healthy overall. But they did improve some aspects of cognition too. Given this, we were really intrigued by this idea then. If individuals exercise, how would that interact with this intranasal insulin? So Dan Batilio, pictured up here, is conducting this for his dissertation, and he's furiously working through data to try and defend. So I have a little bit of data to share, but we designed a protocol where we wanted to assess the single bout effect of exercise. Thinking in the short term, what would the immediate effect of this intranasal insulin do to the body, and how would exercise interact with that? We had people come in, either do exercise at a moderate to vigorous intensity for 60 minutes, meeting Institute of Medicine guidelines for obesity care, and also then do a counterbalance condition where they were randomized to do either one first, and whatever one they didn't do, they would do the second. So if they exercised first, they would then rest second. We then had them come back about 16 hours later to our brain imaging center, where they conducted a series of steps, one of which included using the NIH toolbox to assess cognition. We did blood work and blood pressure assessments, and we used MRI to assess cerebral blood flow, particularly in the hippocampus and the accumbens, given the accumbens has some ties to reward centers. We gave people then intranasal insulin, which importantly, we administered this at 40 IUs. I bring this up because this is considered by many to be an effective clinical dose. Higher values can be supraphysiologic and actually create spillover effects into the periphery such that outcomes are going to be influenced then by peripheral insulin concentrations. So we had provided this. We had then repeated blood draws 30 and 90 minutes later, and we had people then go back into the scanner and repeat measures at about 30 minutes to get some interesting outcomes. We also provided an oral glucose tolerance test, and then we made measurements for about 2 hours. I'm not going to show any of these data for time, and instead I'm going to focus in on this initial period. So some preliminary data. The A and B columns here represent no exercise. And in top here, we're looking at the accumbens. And I'm breaking this into people with normal glucose tolerance and people with hyperglycemia, because our inclusion criteria allowed people with obesity. And we only excluded people if they were on insulin therapy. So we actually had about 8 people who were considered pre-diabetic. When we broke them up, what was interesting to us is actually we see a significant difference. Again, small sample size here, but just in the resting condition, no exercise, that when people receive intranasal insulin, their cerebral blood flow goes down compared to the hyperglycemic group where it goes up. And this was seemingly true in the accumbens as well as a trend in the hippocampus. We then had those people, again, exercise. And if you notice here, we saw an opposite response, where exercise actually lowered cerebral blood flow in responses to insulin in the hyperglycemic group, but not the normal glycemic group. And this was true in both regions with trends occurring. That was really intriguing to us as it actually matches Coleman's group, where they see in the overweight individuals who did not have any hyperglycemia that they increased areas in the putamen. That's what we're seeing in our normal glucose group, but not the people with hyperglycemia. We looked at our insulin signaling with AKT in particular because of the prior findings, and we see something interesting, again, where there's a divergence in response. The normal glycemic individuals have a positive response, meaning insulin activation of AKT was higher, where it was lower in the hyperglycemics. Upon exercise, there was a change again occurring. We were then interested in thinking, well, does baseline fitness matter here? What did their fitness levels do? And could some of the parameters like heart rate recovery, which is commonly used to assess parasympathetic activity, be used in a way to assess this? And looking at that, we end up finding relationships with the neuronal insulin signaling where the people with higher fitness levels through VO2 max tests actually had greater stimulation of both IRS1 as well as AKT. And with heart rate recovery, we want to see a greater decline in heart rate following a maximum test. So we see something similar here, where a greater decline in the heart rates were related to greater stimulation at the level of IRS1 and AKT. And another piece of data here from the vasculature is somewhat following suit, we think, whereas the normal glycemic individuals are having a differential response without exercise in response to that intranasal insulin, where they have a greater blood pressure indice. And this was actually shown recently by Jackie Lindberg's group at the University of Missouri in lean young individuals. In response to intranasal insulin, they had a robust response in blood pressure, but they also had increased muscle sympathetic nervous system activity. When we exercise, we see somewhat of a reversal of that blood pressure response, but now the hyperglycemic individuals in some ways may have a restoration of that response and blood pressure goes up. So we're still trying to figure out exactly what this means. But to say here overall, brain insulin resistance at least seems to be gaining some interest as a therapeutic target, and early work seems very promising. Exercise does seem to have some impacts in the brain, but we certainly need a lot more work as it particularly relates to the phenotypes. And it might be difficult to look at everyone in aggregate. And this idea that the peripheral vasculature might be a really important piece in connecting some of the mind-body interactions. As was also highlighted in a nice symposium earlier today. So understanding the exercise benefits are quite important. And I'd be remiss just to say, if I didn't acknowledge many people in the lab for this, their effort in the study, particularly Dan in that. So with that, I thank you for your time and attention, and I'm happy to take a question or two if we have the time.
So, hi, Steve. Jane Roosh, Denver. I really like that. I kind of love the way the whole thing wove together. And lately we've been trying to, so I'm wondering, because you've mentioned sympathetic tone, you've mentioned peripheral flow, you've mentioned brain flow. And I'm wondering what you're thinking about the metaboreflex in this whole situation. I think that's where we're coming to with our work that we'll talk about later today. But because you're talking about the brain, insulin, increased sympathetic flow, it's essentially the metaboreflex that you would get stimulated with an exercise challenge. And that is disrupted in people with diabetes, so it kind of aligns here. But have you guys been thinking about that, or should we think about it together?
Yeah, I would love to think about that with you, Jane. I'd be a fool if I didn't. So I'll say that to start. We have been. We've been intensively thinking about this in relations of peripheral metabolism and linkages to the brain, and how either peripheral blood flow, carbon dioxide levels, oxygen could be linking into this. So we have some preliminary data where we have looked at the same individuals who have undergone some of these protocols were willing to do a clamp, and we find some relationships between the periphery during peripheral insulin infusion and what we're seeing in the brain in terms of these neuronal EVs. But much more than that, we have not made any specific measures of sympathetic activity or anything of that way.
Yeah, thank you.
Lisa Chow, University of Minnesota. I was surprised by the discrepant response between the normal glucose and high glucose. I was wondering if the baseline was different and what you're seeing is just sort of normalization to the mean.
No, I'm really glad you asked that, Lisa. So again, small sample size. And I think historically, to say in the literature, that's what is proposed in these models when looking at cerebral blood flow. Most times, I believe, when people are making that measurement, whether it's through transcranial Doppler, looking at that middle feeder artery, or looking at the regions of interest, they're only looking at the fasting state. And in those situations, generally, they're either no difference or they're lower. In our case, I think partly with the sample size, again, taking that into consideration, we're not seeing differences. But to your idea, we also have looked at some relationships where those individuals who are responding to insulin initially, so like our hyperglycemics, they're the same people who are getting improvements in the fasting state after exercise. So we do think there's some sort of interaction going on. And when looking at the Coleman article, that's what they see too. So teasing that out is a little tricky, and I think on some levels, there's almost a flexibility of the brain, if you will, that we're hypothesizing is impaired initially, and exercise might be restoring the flexibility and being able to shift states accordingly.
Thank you.
Hi, thank you so much for the amazing talk. My name is Shu Hao. I'm from Mayo Clinic. This may be a little bit off topic, but I'm just interested in if insulin sensitivity for brain, especially, can affect the appetite regulation at all. Do you know anything about that?
I know a little bit. Not as many as probably some in this room, for sure. But we think so. Just in general, insulin, we think of it as an anorexic hormone. Many people have shown that. We do have assessments of appetite, so we've used visual analog scales during the OGTT in particular. So that's some of the work that we're looking into, and then we have blood samples that we were thinking based on that to look at things like ghrelin and GLP-1 to see if there might be some vagal activity that might be interacting. But we haven't analyzed the blood samples, and as I mentioned, a lot of this is still preliminary. We're working through those data, and I hope by the end of the summer going into next year, we'll have some of that insight, but possibly. And that's why with the accumbens, we were interested because of reward centers and feeding. As I'm sure you're familiar with, David Ludwig has, kind of proposed some of the carbohydrate models, and it's kind of fascinating with carbohydrate-based diets. It does seem to activate the the accumbens a bit more with cerebral blood flow measures compared to if you were on lower carb diets. So I think there's something to be figured out there for sure.
Interesting. Thank you so much. I look forward to your results.
Thank you for the question. And maybe this will be the last question. As I know right now, I'm standing between you and the networking social where there's food and beverage. So hopefully many of you will move over there so we can keep the dialogue going.
Yeah, hi, Zoe Urbanitakis from the Rush Alzheimer's Disease Center here in Chicago. We've measured brain proteins in persons who are well characterized during life and come to autopsy at time of death. And we have found that AKT is really important for cognition in people with and without diabetes, but this is measured in human postmortem brain. So it kind of ties in nicely with your data, but there's important differences, and I'm just, what my question is to you is, it seems to me that you're studying relatively young and relatively healthy individuals compared to, say, somebody who, dies at the age of 80 with multiple comorbidities and complications from the diabetes and, on multiple medications, et cetera. So it's much more complicated at that time of life. So my question is, do you think that there's any difference over the life course in terms of what you're finding? Because, we're dealing with a very different population who gets the Alzheimer's disease later in life. You're studying much younger people who don't have AD, ADRD in the brain. So I'm just, what have you thought about in terms of the decades of life that you're studying, and do the effects change with age?
Yeah, great question. So a lot to unpack. I'll do my best. I think part of the reason we chose the age population was middle age. Some might say is actually the impetus and the beginning stages of really when all this is developing, such that later on, to your point, the complicated interactions of hyperglycemia, oxidative stress, inflammation, so forth, there could be multiple mechanisms into what's going on. Whether they're healthy or not, I appreciate that point. At least in this way, part of the reason we broke it up is because of this notion of the pre-diabetic response that we were seeing that was different. That all said, I think based on some work that Demetrios has actually conducted as part of the Baltimore longitudinal trial, they've shown that these neuronal EVs are somewhat predictive of age-related declines in cognition. It doesn't directly answer your question, but I think it might give some insight into something earlier on may actually be changing that is showcasing itself before something like amyloid beta that might reach clinical levels where we're seeing it. And that's why, in part, we're thinking insulin could play roles, given that it does have some implication before that. But I think it's a really interesting question. I think it becomes really complicated. But we're interested in this idea of insulin signaling, and I am glad it matches up with some of the postmortem brain results too. So, thank you.